Welcome to the Deep Dive. We're here to take your source material, really dig into it, and pull out the essential knowledge you need with all the context.
Exactly.
Today, we're tackling Semax. It's this synthetic peptide, and honestly, it's popping up everywhere in conversations about nootropics, about brain repair. Lots of curiosity there.
It really is, and it's got this very specific history stretching back decades, mostly rooted in Russian science. We've got a good stack of sources, detailed molecular stuff, clinical reports from Russia, and importantly, some pretty stark regulatory warnings.
Right. So our mission really is to give you the full picture.
Yeah.
The chemistry, how it supposedly works, what the research actually says, the good, and the limitations.
And we need to jump straight into the most critical piece of context right at the beginning. Based on these sources, it's about safety and regulation.
Absolutely crucial. We have to be crystal clear on this from the outset. Semax, when you see it sold by research chemical suppliers, comes with a very explicit warning.
It does. It's fundamental. These companies state very clearly that it's strictly for research purposes only. That's the label, that's the legal standing outside of its approved medical use in specific countries.
And that means, let's just spell it out, it is absolutely not intended for human consumption. Full stop.
Yeah.
This isn't a suggestion. It's the regulatory reality we have to operate within for this whole discussion.
Precisely. So when we talk about exciting results in, say, animal studies or stroke trials over in Russia, you need to remember that's happening under controlled research conditions or as a prescribed medicine. It's not a supplement. It's not FDA approved in the U.S. or most places.
Okay, got that essential safety framework locked in?
Good.
Because now we can get into the really interesting stuff, the molecule itself. How is this thing built?
All right, the chemistry. Semax is what we call a synthetic heptapeptide. Fancy term, just means it's a chain made of 7 specific amino acids linked together.
7 amino acids, okay.
Yeah.
And is the sequence important?
Absolutely critical. The sequence is the molecule, functionally speaking. It's methionine, glutamine, histidine, phenylalanine, proline, glycine, proline, or medgluhis-fuproglipro for short.
Okay, medgluhis-fuproglipro. Got it. And you said synthetic, so it wasn't just found somewhere.
No, it's engineered, but it's not completely out of the blue. It's an analog, basically a modified version of a small piece of a natural hormone.
Ah, right. This links back to ACTH, doesn't it? Adrenocorticotropic hormone. Why pick a piece of that? ACTH does a lot of things in the body.
It does. Big systemic effects, especially triggering cortisol, the stress hormone from the adrenal glands. But researchers figured out way back, maybe the 50s or 60s, that a tiny fragment of ACTH, just the amino acids in positions 4 through 7, seemed to be responsible for some of its cognitive effects.
But without the big cortisol hit.
Exactly. They wanted the brain benefits without the body-wide stress response. So they isolated that core cognitive piece, the ACTH 4 to 7.
And Semax builds on that.
Right. Semax takes that ACTH 4 to 7 fragment and then, crucially, adds a 3 amino acid tail. Proline, glycine, proline. Proglipro. So you sometimes see it described as ACTH 410 plus that PGP extension.
Okay, so what does adding that proglipro tail do? Why bother?
Ah, that's the clever bit. That tail is all about stability and delivery. It does 2 main things. First, it makes the whole peptide much, much more resistant to being broken down by enzymes in the body, peptidases.
Like giving it armor plating.
Kind of, yeah. So it survives longer after you administer it, whether that's nasally or by injection. Second, this resistance dramatically increases its bioavailability, how much actually gets into circulation and its half-life, how long it sticks around. More gets to the brain, and it stays active longer.
Okay, that makes sense.
Yeah.
Smarter design. Now, the history, you mentioned Russia. This is very much a Russian development, isn't it?
Oh, absolutely. Developed at the Russian Academy of Sciences. The early work on ACTH fragments was happening from the 50s, but the focused effort to make Semax itself, the stable neuroactive version, really ramped up in the 1980s and into the 90s.
And the name Semax. Any meaning there?
Yeah, it's actually pretty neat. It comes from the Russian phrase for 7 amino acids. Sem aminokislot. Reflects its structure.
Simple enough. So this isn't some brand new, untested thing. It's got decades of research behind it over there.
Decades. And that led to its approval. By the early 90s, Semax was approved as a prescription medication in Russia, and I believe some neighboring countries too. It's used primarily for neurological issues, stroke recovery, or some cognitive disorders.
Which creates this really interesting split, doesn't it? A prescription drug in one part of the world and strictly a research chemical almost everywhere else.
Exactly. That's the landscape we're navigating.
Okay, so this engineered chain of 7 amino acids gets into the system, maybe via nasal spray. How does it actually start influencing the brain? What's the mechanism? We know it's probably not just one simple thing.
No, definitely not simple. It's more of a neuromodulator, meaning it influences several systems at once. But the biggest, most well-documented effect, the one everyone points to, is its ability to significantly boost levels of BDNF.
Brain-derived neurotrophic factor,
Right.
often called miracle grow for the brain because it helps neurons grow, survive, make connections.
That's the one, essential for neuroplasticity, learning, memory. And Cmax is a potent BDNF booster.
Where does it boost it? And how fast? Is this a slow, long-term thing?
That's what's really striking. It targets key areas, the hippocampus, crucial for memory formation, and the basal forebrain, important for learning and attention. And the speed is remarkable. Animal studies show intranasal Cmax jacks up BDNF protein levels and the levels of its receptor, TRKB, within just a few hours.
Hours. Wow. Okay, so that connects directly to potential cognitive effects quite quickly then. It's not just about slow growth over weeks.
Exactly. It suggests an acute effect potential, not just long-term maintenance. It seems to optimize the existing machinery pretty rapidly.
Okay, BDNF is the big one.
Hmm.
What else is going on? You mentioned influencing systems, neurotransmitters.
Yes. The sources indicate it modulates or kind of fine-tunes both the serotonin and dopamine systems simultaneously.
So mood, motivation, attention, alertness, all that complex stuff.
Pretty much. By activating these pathways, it seems to enhance the signaling related to all those functions. It's like improving the efficiency of the brain's communication network for tasks requiring focus and stable mood.
I saw a note in the materials. It said Semax can potentiate stimulants, make them stronger, but doesn't really raise dopamine levels itself. How does that work?
That's an important distinction. It doesn't seem to act like, say, amphetamine, which directly causes a flood of dopamine release, or like an SSRI, which blocks reuptake. Instead, it might make the dopamine receptors more sensitive or make the existing dopamine signaling more efficient somehow.
Ah, okay. So it's enhancing the effect without necessarily just cranking up the volume.
That seems to be the idea. It's a more subtle modulation, which might explain why it doesn't seem to have the same addiction profile as direct dopamine agonists or releasers. Cleaner, perhaps.
Right. Let's get down to the receptor level. Since it came from ACTH, it must interact with those melanocortin receptors, right? MC4 and MC5.
It does. The melanocortin system is involved in a whole range of things: energy balance, alertness, inflammation, cognition. Cmax binds to MC4 and MC5 receptors. The thinking is it might act as a partial agonist or antagonist there, basically fine-tuning their activity.
Okay, but crucially, it avoids the main ACTH receptor, doesn't it? The one that triggers cortisol.
That's the absolute key to its design. It does not activate the MC2 receptor, which is the one in the adrenal cortex responsible for pumping out cortisol. So you get that a potential cognitive and neuroprotective actions linked to MC4, MC5 without the body-wide stress cascade from MC2 activation. That was a major goal of the synthesis.
Very clever. Okay, one more piece of the mechanism puzzle. Inhibiting enkephalinases. What on earth are those?
Yeah, sounds complex. Enkephalatase are simply enzymes. Their job is to break down the body's own natural opioid peptides, the enkephalins.
Are internal painkillers and mood regulators.
Exactly. Things involved in managing pain, stress response, mood. By inhibiting the enzymes that chew up these enkephalins, Cmax might allow them to stick around longer or reach higher concentrations.
So potentially another route to influencing mood and resilience.
Potentially, yes. It's maybe a secondary mechanism compared to the BDNF boost, but it fits with some of the observed anti-stress effects we'll talk about. The biological significance is still being fully worked out, though.
Okay, so summing up the mechanism. Big BDNF boost, tuning up serotonin and dopamine, targeted melanocortin receptor action without the stress hormone, and maybe preserving some natural mood regulators. It's multifaceted.
Very much so. A complex neuromodulator.
All right, we've got the how. Let's move to the what. What does Cmax actually do according to the research? Let's start with the big one. Cognitive enhancement. Does it actually make animals or people smarter?
Yeah.
#TagNAA. Cognitive enhancement and learning.
Well, in animal models, the evidence is pretty strong and consistent over many years. The sources are clear. Cmax reliably improves cognitive performance in tasks related to learning and memory.
Can you give us a concrete example, something that really shows it working in the lab?
Sure. A classic test is the conditioned avoidance response. Basically, you train a rat to avoid a certain place or action associated with something unpleasant, like a mild shock. Rats treated with Semax learned this avoidance task significantly faster and remembered it better over time compared to the control group.
So faster learning, better memory consolidation.
Exactly. Or maze tasks running through a maze to find a reward. Semax-treated rats often completed the mazes faster and with fewer errors, showing improved spatial learning and memory retention.
Okay, that's compelling in animals. We also talked about that rapid effect, some studies showing improvements just 15 minutes after a dose. How does that square with BDNF, which sounds like it should take longer to build new connections?
That's a really good question. The very rapid effects, like within minutes, are likely due to two things happening at once. First, the immediate modulation of serotonin and dopamine that's fast-acting, like flipping a switch to improve signaling efficiency.
Clearing the static, improving focus right away.
Pretty much. Second, while the physical growth from BDNF takes time, the signaling cascade starts immediately. The levels of BDNF protein and its receptor TRKB start rising quickly, which likely optimizes the cellular environment for learning and memory formation almost instantly, even before major structural changes occur.
So the quick hit is neurotransmitter tuning plus the start of the BDNF surge, preparing the ground. The longer-term benefit is the actual rewiring supported by sustained BDNF.
That seems to be the most plausible explanation, yeah. And crucially, studies have shown a direct correlation. The better performance in these cognitive tasks lines up with measured increases in hippocampal BDNF and TRKB levels. The mechanism and the outcome are linked. ###B neuroprotection in acute injury, stroke.
Okay, let's shift gears to where Semax has actual improved clinical use stroke. It's prescribed in Russia for ischemic stroke and TIAs, right? What's the evidence that actually protects the brain during that kind of crisis?
This is probably the area with the strongest clinical and preclinical backing. Stroke creates a brutal environment in the brain, lack of oxygen or hypoxia, and this flood of glutamate that overexcites and kills neurons, called excitotoxicity. Cmax appears to act as a cellular bodyguard in that situation.
How does it protect the cells?
It seems to help maintain cellular energy balance. Mitochondria, the power plants of the cell, often collapse during a stroke. Cmax helps stabilize mitochondrial function, keeping the energy supply going. It also seems to directly counter some of the damaging effects of low oxygen and high glutamate.
And does that protection translate into less brain damage overall?
Yes. Animal studies consistently show that giving Cmax after an induced stroke significantly reduces the size of the infarct, that's the area of dead tissue. That's a key measure of neuroprotection, and Cmax delivers on that in these models.
Now here's where it gets really interesting for me. The sources mention genome-wide studies showing Cmax doesn't just protect, it actively changes gene expression after a stroke. What's going on there?
This is fascinating. It suggests Cmax isn't just passively shielding neurons, it's actively orchestrating a repair response. These studies looked at which genes get turned on or off in the hours after a stroke when Cmax is given.
And what does he find?
They found Cmax dramatically ramps up the expression of genes involved in two critical repair processes, the immune response and angiogenesis, which is the formation of new blood vessels.
Wow, so it's basically telling the brain, okay, manage the inflammation properly, clear out the debris, and start building new pipelines to restore blood flow.
That's a great way to put it. For instance, they saw increased expression of things like chemokines, signaling molecules that guide immune cells and immunoglobulins within the first day post-stroke. And the boost in angiogenesis genes suggests it's priming the damaged area for vascular repair, which is absolutely vital for long-term recovery.
And this isn't just in rats, this ties into the clinical use.
It seems to. Clinical trials in Russia have recorded that adding Cmax to the standard stroke treatment protocols leads to better neurological recovery scores in patients. And interestingly, they've even measured higher levels of circulating BDNF in stroke patients receiving Cmax during rehabilitation. It connects the dots from mechanism to outcome. #C. Mood, stress, and resilience.
Given the links to serotonin, dopamine, and maybe those enkephalins, it makes sense to ask about mood and stress. Does the research show Cmax helps buffer against stress or improve mood?
In animal models, yes, quite consistently. Studies report antidepressant-like and anxiolytic-like, meaning anxiety-reducing effects.
How do they test that in rats? You can't just ask them how they're feeling.
Yeah, no. They use standardized behavioral tests. For example, they might put rats under conditions of chronic stress, like unpredictable noise or confinement. Control rats under this stress typically lose weight, show high anxiety behaviors like freezing or avoiding open areas.
And the Cmax rats.
The rats receiving Cmax during the chronic stress period show significantly less anxiety behavior. They explore more, they don't lose as much weight. It suggests Cmax increases their resilience, their ability to cope physiologically and behaviorally with the ongoing stress.
Like a stress shield, almost.
Sort of. And mechanistically, this ties back nicely to the BDNF boost and maybe NGF nerve growth factor, too. We know chronic stress is physically damaging to neurons, especially in areas like the hippocampus. By boosting these growth factors, Cmax might literally be helping neurons resist or repair that stress-induced damage.
So it's potentially building resilience at a cellular level.
That seems to be the idea. It aligns with the core mechanism. Now, translating that to humans, some users anecdotally report feeling more alert, maybe less anxious, which fits the neurotransmitter modulation. But formal studies on mood and stress in healthy humans are pretty limited. #TAG-T neurodegeneration, theoretical interest and limitations.
Okay, what about the really big chronic neurodegenerative diseases? Parkinson's, Alzheimer's, any hope there?
This is where we need to be really cautious and manage expectations. For Parkinson's, the animal data is quite mixed. Some studies using toxin models like MPTP, which mimics some Parkinson's damage, showed maybe slight improvements in motor function with Cmax, but another study found it actually increased anxiety in that same model. So not exactly a clear win. It's not seen as a promising therapy for Parkinson's motor symptoms right now.
Right. Mixed signals at best.
Yeah.
What about Alzheimer's? There seems to be some theoretical interest there, something about copper.
Yes, this is an interesting angle, but purely theoretical at this stage, mostly based on lab dish studies. Alzheimer's involves the buildup of amyloid beta plaques. It turns out that copper ions, which are naturally present in the brain, can bind to these amyloid peptides and make them clump together faster, and also generate damaging free radicals.
And Cmax interacts with copper.
In vitro in the test tube, yes. Studies have shown Cmax can chelate copper ions.
Chelate, like grab onto them.
Exactly, like a molecular claw. By binding the copper, Cmax potentially prevents it from speeding up amyloid plaque formation and generating those free radicals. Lab studies showed it could inhibit the formation of these toxic amyloid copper complexes and slow down the growth of amyloid fibers in artificial settings.
That sounds potentially huge for an amyloid disease. So why isn't everyone talking about Cmax for Alzheimer's?
Because what happens in a petri dish often doesn't translate to a living brain, especially with something as complex as Alzheimer's. Despite that intriguing in vitro finding and the known BDNF boost, the overall scientific consensus, looking at recent reviews, is pretty blunt.
What do they say?
They essentially say there's not a strong rationale and little evidence to suggest that nasal Cmax is actually helpful for age-related cognitive decline or dementia in humans. We simply don't have well-controlled clinical trials showing it slows disease progression. The theory is interesting, the reality isn't there yet.
Okay, a very necessary dose of reality. Success in acute injury like stroke doesn't automatically mean it works for slow chronic degeneration.
Yeah.
Different ballgame.
Completely different. #TAG-V synthesis, lab use, and regulatory context.
Let's shift to the practical side for researchers. How is this stuff actually made? Metgluhisfe-Pro-Glypro. You can't just whip that up easily, right?
No, definitely not. It requires highly specialized chemical synthesis. The standard method is called solid phase peptide synthesis, or SPPS. It's the workhorse for creating custom peptides like Semax accurately.
Solid phase. Can you walk us through that briefly? How does it ensure the sequence is right?
Sure. You start with tiny insoluble resin beads. The last amino acid in the chain in this case, proline, gets chemically anchored to these beads. Then you sequentially add the next amino acid, glycine, then the next, proline, and so on, working backwards along the chain. Baphihes, glue, met.
One amino acid at a time, sticking them onto the growing chain on the bead.
Exactly. Each step involves carefully controlled chemical reactions, protecting the parts you don't want to react, activating the parts you do, adding the next amino acid, washing away excess stuff, then deprotecting for the next round. It's meticulous step-by-step construction.
Okay. Once the full 7 amino acid chain is built on the bead, how do you get the finished Semax off?
You use a chemical cocktail, often strong acids, to cleave the completed peptide off the resin bead. Now you have your crude Cmax, but it's mixed with leftover regions, maybe some failed sequences. It needs cleaning up.
Purification. How critical is that?
Absolutely critical for research. You need to know you're testing Semax and not some contaminant or shorter inactive fragment. The standard purification method is high-performance liquid chromatography, HPLC. It separates the target peptide from impurities based on chemical properties.
The researchers need really high purity, right? Like 99%.
Ideally, yes.
Yep.
Research-grade peptides often aim for 98% or higher purity, confirmed by HPLC and sometimes mass spectrometry, just to be sure about identity and concentration. Anything less, and your experimental results could be meaningless or misleading.
Makes sense. Once it's pure, how is it stored? It sounds like a delicate molecule.
It is. It's typically sold as a lyophilized powder freeze-dried. This makes it relatively stable for shipping. But for long-term storage in the lab, you keep it frozen solid, usually below -Celsius, so like a standard lab freezer.
And dissolve it when needed.
Right. When you're ready to use it, you reconstitute the powder in sterile water or saline to a known concentration, say 100 micrograms per milliliter, depending on the experiment.
In getting it into the animal or system,
Yeah.
we know oral doesn't work.
Correct. Oral bioavailability is practically zero stomach acid and enzymes just shred it. So in research and in its clinical use, it's almost always intranasal spray, which offers relatively direct access to the brain via olfactory pathways, or injection like subcutaneous or intramuscular.
Okay. Now, we focused heavily on the brain, but the sources mentioned Semax pops up in other research areas too.
It does, which speaks to its broader biological effects. Because it modulates inflammation and vascular growth, angiogenesis, it's used as a tool in immunology research looking at neuroimmune crosstalk.
Interesting. Anywhere else?
Yeah, ophthalmology researchers are exploring if it can protect retinal neurons or optic nerve fibers in models of glaucoma or eye stroke. And even gastroenterology, there's some work looking at its effects on gut blood flow and healing peptic ulcers, potentially linked to its vascular effects.
Wow. So it's really a versatile molecular probe, not just a brain drug.
Definitely. Which brings us back, full circle, to the regulatory context.
All right. Let's hammer this home one last time.
Outside of its specific approved medical uses in Russia and a few other places, the label you'll find from suppliers is unambiguous, for laboratory research use only. It might be unscheduled in many countries, meaning not strictly illegal to possess, but that does not mean it's approved or legal for human use as a drug or supplement. That research-only status is the key boundary.
Got it. Research tool. Period. Hashtag tag Zsong's outro.
So wrapping up this deep dive on Semax, it's clear this is a fascinating peptide with a rich history.
Yeah, definitely not simple. Let's recap the big takeaways for everyone listening.
Okay. We saw it started as an engineered fragment of ACTH, that 7-amino acid chain metgluhis-phi-pro-glypro, specifically designed to keep the cognitive bits but ditch the systemic stress response.
And its main mechanism seems to be a really potent, quite rapid boost in BDNF, that crucial brain growth factor, especially in memory centers like the hippocampus.
Plus, it fine-tunes serotonin and dopamine and maybe helps preserve natural mood regulators.
Where it shines, based on the evidence, is in acute neuroprotection, particularly stroke. The animal data showing reduced infarct size is strong, and the clinical use in Russia supports that. And that link to boosting genes for immune response and blood vessel repair post-stroke is really compelling.
Super compelling. But then there's the crucial counterpoint. Despite interesting lab dish findings like chelating copper relevant to Alzheimer's theory, there's just no solid clinical evidence right now that it helps with chronic neurodegeneration like Alzheimer's or dementia in humans. The leap from theory to therapy hasn't been made.
And overriding everything is the regulatory reality. Outside of those specific Eastern European medical approvals, it remains strictly a research chemical, not for human consumption. That cannot be stressed enough.
Absolutely.
Okay, so let's leave you with a final thought to chew on, building on that gene modulation piece.
Right. We talked about how those genome-wide studies revealed Cmax flipping switches after a stroke, actively turning on genes for vascular repair, angiogenesis, and coordinating the immune cleanup. It's not just protecting, it's initiating rebuild.
Which really makes you wonder, does the brain have this incredible latent capacity for self-repair, especially in terms of rebuilding its own blood supply and managing inflammation, that we haven't fully tapped into?
Exactly. If this relatively simple peptide can kickstart those specific repair pathways so effectively after an acute injury, what does that tell us? Could future research design even more targeted molecules? Maybe next-gen peptides, maybe small molecules that leverage those same vascular and immune repair mechanisms even better?
Moving beyond just stroke recovery, perhaps, to tackle chronic conditions where vascular health and inflammation are key problems.
That's the provocative question, isn't it? How do we harness those endogenous repair systems more effectively? Semax kind of gives us a glimpse into activating them.
A glimpse into a potentially powerful future direction for research. Definitely something to think about.
Keep thinking, keep questioning,
and keep diving deep with us. Until next time.